Alright, today we'll be discussing enzyme inhibition, and I will go through a series of introductory slides that will explain why is it important to be able to inhibit enzymes, what types of characteristics are preferred in an enzyme inhibitor, and then as time permits I will cover two case studies of enzyme inhibitors, and I will probably have an additional lecture on a different day on other case studies that I would like you guys to have. Okay, so let's begin our conversation today on enzyme inhibition. So first of all, here I'm showing you some structures of enzymes and the enzymes that are some structures of enzyme inhibitors, the names of the enzymes that are inhibited, and the class of enzymes that are inhibited here. Dasatinib is an anti-cancer. Pazopanib is an anti-cancer as well. Aliskiren is an antihypertensive, and saxagliptin is anti-diabetic, is a dipeptidyl 4 inhibitor. Dipeptidyl 4 inhibitor. Okay, so why do we need to inhibit an enzyme? Sometimes you need to inhibit an enzyme because you have, let's say, a cell has a deficiency in a particular substrate. And the fact that the cell has a deficiency in this particular substrate leads to a particular disease or a particular condition. So you want to inhibit the enzyme that's actually going to catalyze or that's going to metabolize that particular substance so that you eliminate the disease. For instance, there is a particular substance called gamma aminobutyric acid. Okay, gamma aminobutyric acid levels, when they are decreased, they lead to seizure. Okay, they lead to seizure. Now, there is an enzyme called gamma aminotransferase. GABA aminotransferase. Okay, and that enzyme is responsible for clearing out gamma aminobutyric acid from the system. So if you have decreased level of gamma aminobutyric acid that lead to seizure, one of the ways to treat seizure and give anticonvulsant effect is to inhibit the activity of GABA aminotransferase. So that's one reason why you want to inhibit an enzyme. Other times you want to inhibit an enzyme because the cell is making too much of a compound. You know, the cell is making too much of a substance, and the excess of that substance is leading to a disease or a condition. For instance, in gout, in the case of gout, gout is caused by increased uric acid levels. Increased uric acid levels. So in the case of gout, what you want to do is you want to find out what's the precursor for uric acid and what enzyme converts that precursor to uric acid, and then you block that enzyme. So the precursor for uric acid is xanthine. Okay, and xanthine is converted to uric acid by xanthine oxidase. Okay, xanthine is converted to uric acid by xanthine oxidase. So one of the ways to decrease uric acid levels and prevent gout flare-ups is to inhibit the enzyme xanthine oxidase. Another way why we may want to inhibit an enzyme is because either a bacteria or a virus has a specific, let's say, a specific protein that is essential to the functioning, to the survival of that bacterial virus, and we would like to inhibit that particular virus or that particular bacteria by inhibiting the enzyme. So we target that enzyme. So for instance, in the case of HCV, HCV has a non-structural protein 5, and there's a drug on the market called sofosbuvir. Probably going to misspell this name, sofosbuvir, I believe that's what it's called, that actually inhibits NS5. And then for COVID-19, you have remdesivir, which is a drug that's actually going to inhibit the RNA-dependent RNA polymerase of COVID-19, and the drug is, I believe it's remdesivir. So those are some of the reasons why we would want to inhibit an enzyme. Okay. Now, enzymes are excellent targets for drug design and discovery because if you understand the mechanism, of the enzyme, and if you know a substrate of the enzyme, you can then design analogs of that substrate, or analogs of that substrates that are actually going to be able to interact, with the enzyme. Okay, so this analogs can either look like the substrate, they can look like the transition state, okay, they can look like the product, and so you have drugs that are designed for every step of each one of these cases here. Okay. Now, when you understand how the enzyme works, you understand the mechanism of the enzyme, you know which amino acids are involved in the activity of the enzyme, then you can also design mechanism-based inhibitors. So inhibitors that are going to inhibit the enzyme because of the type of mechanisms that they have. So some of the properties of enzymes that make them attractive is the fact that they can be specific to a target. Okay, for instance, the non-structural protein 5 of HCV is specific to HCV. It's different from the human proteins, and so there is a certain level of selectivity there. It can also be dose dependent. Sometimes you don't have the selectivity between humans and either the pathogen or the disease condition that you're trying to treat, but it could be dose dependent. So for instance, in the case of cancer treatments and you're trying to inhibit an enzyme, sometimes inhibiting a particular enzyme in cancer cells is also going to affect normal cells, but you have a dose dependency there, okay, dose dependency, but you can also have a kinetic effect, because in the case of cancer you have rapidly dividing cells that are going to be affected, so you can actually administer a drug that's going to specifically target those cancer cells because the cancer cells are more rapidly dividing, and then you can inject a rescue antidote to make sure that the medication that's killing the cancer cells is not also killing the normal cells. So when I talk about specificity, for instance, I always go back to penicillins. Okay, so penicillins are drugs that specifically inhibit bacterial bacterial cell wall synthesis. Bacterial cell wall synthesis. Okay, and they do that by inhibiting the peptidoglycan transpeptidase. So penicillins are non-toxic. For the most part, they're non-toxic because the only way they'll be toxic to you is if you are actually allergic to penicillins, and this happens. I think the last thing I want to mention maybe as far as enzymes is that we can group them in two big groups. We can group them in the reversible inhibitors and the irreversible inhibitors. Okay, most of the enzyme inhibitors that we have on the market will be reversible, and these usually have non-covalent bonding interactions. The irreversible inhibitors often have covalent bonding interactions. Okay, covalent bonding interactions. So we will go through some examples here, and I will try to walk you through as much as possible. So when we talk about reversible, if I can just draw a straight line. So when we talk about reversible inhibition, you remember when we were talking about an enzyme and the substrate, right? The enzyme can have a enzyme substrate complex. The enzyme can form an enzyme substrate complex that's then going to lead to the formation of a enzyme product complex, and then the liberation of a product. Okay, so you have that. But the same way, you can also have a enzyme inhibitor. You can have the enzyme inhibitor, and you form an enzyme inhibitor complex. Okay, and so let's do it this way. So in the presence of the substrate, and this is, and then you remove the substrate here. Okay. There's an equilibrium constant. So if this is K on and K off, the equilibrium constant Ki is going to be equal to K off over K on. Okay, so it's a dissociation constant. It's association constant of the enzyme inhibitor complex. So the smaller the Ki, the more potent the inhibitor. The smaller the Ki, the more potent it is. Okay. Now another value that's important for us to know is the IC50. Let me just go up here. I'm running out of space. Well, maybe I can do it here. The IC50. Okay. So the IC50 is another common value to rank the potency of an enzyme. So the IC50 is the inhibitor concentration that produces 50% of enzyme inhibition. Okay. So in inhibitor concentration that produces 50% enzyme inhibition. Now, one thing about the IC50 that I did not know when I was when I was a student is that the IC50 is dependent on the substrate concentration. So if you are going to actually look at, if you're going to compare two IC50 values between two inhibitors, you have to make sure that they're using the same substrate concentration for the assays. Otherwise, those values cannot be related. Okay. Now there is also a way to relate the IC50 to the Ki, and if IC50 is equal to 1 substrate concentration over Km times Ki. Okay. All right. Let me see here. So, other things that I would like to mention here, as I'm erasing this side of the board, we can also talk about the same way we talked about reversible, irreversible. We can also talk about competitive and non-competitive inhibitors. Okay, competitive, non-competitive inhibitors. All right. So when we talk about competitive inhibitor, let me change the color of the pen just for the sake of it. Okay. So when we talk about competitive inhibitors, competitive inhibition in the case of competitive inhibition, you actually have the inhibitor competing with the substrate. So the inhibitor is going to bind at the same place where the substrate binds. Okay, so it's either going to block access to the active site, or it can also remove the substrate from the active site. Okay, so it can simply block and not be transformed by the enzyme. Okay, or it can actually react with the enzyme and then produce what we'll call useless metabolites. Okay. Now the issue with these useless metabolites is they can actually be toxic. So that's something to keep in mind. Now, non-competitive competitive inhibition is what we covered when we mentioned allosteric sites. Okay, allosteric sites. Okay, so basically the inhibitor is going to bind at an allosteric site, causing a conformational change, preventing the substrate from binding to its enzyme. Okay, so let me give you some case studies here. The first case study I want to look at is a group of protein kinases, and especially the let me see what I have in my notes here. The epidermal growth factor receptor. Okay, epidermal growth factor receptor. Now, this is the epidermal growth factor receptor. Okay, so it's a receptor it's a transmembrane protein that has on one side of it the receptor, and on the other side it has the kinase domain. So protein kinases catalyze the transfer of the terminal phosphate group of adenosine triphosphate ATP to the hydroxyl group of serine, threonine, tyrosine residues of protein. Okay, so that's what protein kinases do. Kinases are present in a lot of different pathways that communicate to cells, and they're involved in cell proliferation. They're involved in cell growth, and so on and so forth. Now, if you have changes, or the big word is aberration, if you have an aberration in specific kinases, this has been shown to lead to cancers and tumors. Okay. Specifically, there's a number of tumors and cancers and conditions that have been connected or that have been related to the EGFR receptor and the kinase activity of the EGFR receptor. So the EGFR kinase inhibitors became a potential way to inhibit the kinase of the EGFR and thus treat cancer. Okay, so let's take a step back and think about it here. So the EGF, okay, the EGF is a polypeptide that binds to the outer layer surface of targeted cell. So it binds to the surface of targeted cell to stimulate cell growth, proliferation, and survival. Okay. Now the receptor itself, that is here, is a transmembrane protein receptor that contains a kinase domain. The possible approaches to developing treatment would be because you have aberrations. So let's say you have an aberration to this particular EGF receptor here, and you don't want the communication to continue inside of the cell. One of the ways you can do it is you can try to block the binding of the EGF to the receptor, and that can be done through antibodies. And there are some drugs on the market for that. Another way to do it is to inhibit the kinase enzymatic function of the receptor. And that can also be done by designing specific inhibitors that are going to specifically bind there and inhibit the activity of those kinases. Okay? So EGFR receptor kinase inhibitors were discovered by scientists at AstraZeneca, and I also want to say at Park. It's another company. And what they did is they did a screening. They did a virtual screening. Okay, and what they were trying to look at is molecules or moieties that mirrored or had similar characteristics as tyrosine. The reason why they were looking at that is mechanistic studies had shown that the transfer of the phosphate group from ATP to the protein was a direct result of a nucleophilic attack of a tyrosine on the gamma position of the phosphorus group of the triphosphate. I believe it's a gamma triphosphate. So tyrosine was the molecule that was going to be used as basically a lead compound. So a few of the consideration is that the molecule that was going to be chosen had to have a 6-membered aromatic ring to mirror um, tyrosine in the substrate. It had to have an oxygen or a nitrogen atom at a suitable distance from the aromatic ring, still to mimic the phenolic hydroxyl group. Okay, and one of the considerations that they added, it had to have a oxygen or a nitrogen at a suitable distance from the hydroxyl group to mimic at least two of the non-bridging oxygen of the triphosphate of ATP. Okay, so this to mimic two of the non-bridging oxygens that are present in the triphosphate. So these are the three considerations that they had. Now they went ahead and ran the screening through a library. They did the virtual screenings through a library of compounds that they had available at the company, and they came up with these molecules here. Okay, they came up with these molecules here. So at the same time as AstraZeneca was doing the screening, the other company also were doing the screening, and they came up with very similar structures, as you can see, very similar structures. So these structures then were improved upon to give gefitinib and erlotinib, which are EGFR inhibitors. So the assumption would be that these drugs were designed based on the structure of tyrosine, and so mechanistically, when we look at the mechanism of action, the mechanism of action should at least reflect or mirror the original intent of the scientists who designed the drug. But as studies were done, it was actually shown that this particular drug, erlotinib, was actually not competing with the tyrosine for binding. It was actually binding where the ATP would bind. Okay, it was actually binding where the ATP would bind, and there is a hydrogen bonding interaction between the methionine and the nitrogen here that you can see in this graph here, that is also present when you have an analog of ATP that is crystallized within the binding pocket of the enzyme. So in conclusion, aberration in the EGFR receptor activity was shown to lead to cancer. So the EGFR receptor became a target. Okay, drugs were designed specifically to inhibit the kinase activity. Okay. Tyrosine structure was mimicked. we end up with an actual inhibitor. It works, it inhibits the EGFR, but it actually competes with ATP for binding. So this is one of these examples where it shows that you can have rational drug design, but sometimes the original idea may not pan out to be exactly the way you want it to be. Now, what's good to remember as well, what's good to know is that the active enzyme, or the enzyme in its catalytic form actually forms a complex between the substrate, it's an enzyme, substrate, and ATP complex. Okay, enzyme, substrate, and ATP complex. And so these EGFRs are still good targets for conditions such as cancer. And I have another example here that I want to show, but it's another kinase. So I'll give you another example of another kinase, and I think that will be enough for this lecture here. This is actually a reversible. It's not irreversible. Let me keep my annotations here. It's not irreversible. I'm not sure why I have this. Okay, is that reversible? Okay, all right. So let's go back to this. So another example is imatinib. Imatinib is okay. First, let's talk about the story of imatinib. Imatinib is a drug that was the original structures for imatinib, as you can see here. Can I go back? Okay, the original structures that gave rise to imatinib were actually being studied for a condition to be inhibitors of platelet-derived growth factors. Platelet-derived growth factors. So these structures here were specifically designed to inhibit platelet-derived growth factors. Now, there is a condition called chronic myelogenous leukemia. And in chronic myelogenous leukemia, you have an excessive proliferation of a specific type of white blood cells in the bone marrow. And this is associated with chronic myelogenous leukemia is associated with a chromosome called the Philadelphia chromosome. Philadelphia chromosome. Okay, so it's a chromosomal abnormality that leads to the expression of an abnormal protein called BCR. Okay, ABL. Okay, now the BCR stands for breakthrough cluster region. Breakthrough cluster region. And the ABL is the Abelson kinase. So in an attempt to treat the chronic myelogenous leukemia, okay, inhibitors of the BCR ABL kinase started to be developed. Okay, so that's what takes us to imatinib. So imatinib was originally being designed as a drug to inhibit platelet-derived growth factors. Okay, platelet-derived growth factors. But then it was tested. Those compounds were tested against the precursors of imatinib were tested against BCR-ABL and found to have actually inhibitory activity on the kinase activity of the ACR-BCL. Okay, so these compounds were then further developed into imatinib. So the interesting thing about imatinib here and the way it works that's different from what we've seen before is that imatinib actually binds to and stabilizes an inactive form of the enzyme. So it binds to and stabilizes an inactive form of an enzyme, and by doing so, it basically blocks the enzyme from being in the active form. So this is the crystal structure of imatinib here, bound to the to the enzyme, and this is the crystal structure of another inhibitor that is also bound to the enzyme. When you look at the structure of the enzymes, you realize that they are very different. This is the active form, and this is the inactive form. Okay, so what imatinib does is imatinib is actually going to bind to the inactive form of the enzyme, okay, and form a complex with that inactive form of the enzyme. And that's how it exhibits its inhibitory activity. Okay, I have a few other case studies that I'm going to be doing. What I will do is I'm probably going to extend the lectures on enzymes for another couple of videos in the coming weeks, and reduce the lectures on receptors to just one series of videos. So I hope this is helpful. Make sure that you do the homework, submit the assignments, participate in the discussion group. Today, quickly, I want to summarize what we've covered. We've covered why enzyme inhibition is important. Okay, we've covered why enzyme inhibition is important. We mentioned that we inhibit compounds that are in excess. Okay, inhibits the formation of compounds that are in excess that cause diseases. We inhibit the removal of compounds that when we are deficient lead to disease. We've talked about reversible versus irreversible inhibitors. We've talked about competitive versus allosteric inhibitors.